हेलो गुड मॉर्निंग डियर फ्रेंड्स। पिछले क्लास में हमने देखा न्यूरोग्लिया और उसके टाइप्स। सो टुडे वी डिस्कस अबाउट क्लासिफिकेशन ऑफ न्यूरोग्लिया। व्हाट इज न्यूरोग्लिया? न्यूरोग्लिया इज सपोर्टिव cells or helping cells of nervous tissue. Neuroglia is divided into two parts, mainly neuroglial cells of central nervous system and neuroglial cells of PNS or peripheral nervous system. So there are two types of neuroglial cells, neuroglial cells of central nervous system and neuroglial cells of peripheral nervous system. Neuroglial cells of central nervous system further divided into four subtypes: ependymal cells, astrocytes, oligodendrocytes, and microglia. Ependymal cells, astrocytes, oligodendrocytes, and microglia. So, ependymal cells are arranged in a single layer. They are cuboidal or columnar in shape, since microvilli and cilia are present on their apical surface. So, character of ependymal cells is it is arranged in single layer. It is arranged in single layer, cuboidal or columnar in shape. Microvilli and cilia are present on their apical surface. Morphology or appearance of ependymal cells are: it arranged in single layer, in shape cuboidal or columnar, and microvilli and cilia are present on their apical surface. This is diagram of ependymal cells. Function of ependymal cells. Ependymal cells are responsible for the formation of cerebrospinal fluid and assist in its circulation. Ependymal cells are responsible for the formation of cerebrospinal fluid or CSF and assist in its circulation. They act as receptors that monitor the composition of CSF. They also transport the dissolved gases, nutrients, waste, and other materials between the brain tissue and CSF. Function of CSF. It is responsible for formation of cerebrospinal fluid and assist in its circulation. They act as receptors that monitor the composition of cerebrospinal fluid. It also transports the dissolved gases, nutrient, waste, and other materials between the brain tissue and CSF. So, function of CSF broadly. Function of ependymal cells is divided into two parts: formation, circulation, and excretion. In formation, it helps in formation of cerebrospinal fluid and assist in its circulation. They act as receptors that monitor the composition of cerebrospinal fluid. They also transport the dissolved gases, nutrient, waste, and other materials between the brain tissue and cerebrospinal fluid. Next is astrocytes. Astrocytes are so called because they are star-shaped. Its appearance is a star-shaped, so called astrocytes. They have many star-like radiating process. Embryologically, astrocytes develop from neural crest. Embryologically, astrocytes develop from neural crests and its appearance is star-like, so it called astrocytes. Astrocytes of two types, protoplasmic astrocytes and fibrous astrocytes. Protoplasmic astrocytes and fibrous astrocytes. Protoplasmic astrocytes mainly found in gray matter. Fibrous astrocytes help in providing nutrients to neuron and also play a role in the movement of metabolites and waste to and from neuron. They also help in formation of blood-brain barrier. Protoplasmic astrocytes found in gray matter. Fibrous astrocytes helps in providing nutrients to neuron and also play a role in the movements of metabolites and waste to and from neuron. It helps in formation of blood-brain barrier. Oligodendrocytes. Oligodendrocytes are small, round or oval cells with only a few cytoplasmic processes. This is oligodendrocytes. Function of oligodendrocytes: it produces myelin sheath around the axons of multiple neurons in central nervous system. It produces myelin sheath around the axon of the multiple neurons in the central nervous system. It also forms a supportive network around the central nervous system. Microglia. Microglial cells are involved in phagocytic activity within central nervous system. Microglial cells are involved in phagocytic activity within the central nervous system. Function. Microglial cells. Functions of microglial cells or microglia. Its phagocytic activity. Microglia can engulf invading microorganisms and clear away dead cells and damaged myelin of central nervous system. Functions of microglia: it engulf invading microorganisms and clear away dead cells and damaged myelin of central nervous system. These cells are highly active in many degenerative diseases like Parkinson's, Alzheimer's and multiple sclerosis. Microglial cells are highly active in many degenerative diseases like Parkinson's, Alzheimer's and multiple sclerosis. Next is neuroglial cells of peripheral nervous system, in which number one is Schwann cells. Schwann cells serve as the myelinating cells of the peripheral nervous system and support cells of peripheral neuron. A Schwann cell forms a myelin sheath by wrapping its plasma membrane concentrically around the inner axon. They play essential role in the development, maintenance, function and regeneration of peripheral nerves. In the mature nervous system, Schwann cell can be categorized into two major classes, myelinating and non-myelinating. Schwann cells play an essential role in the development, maintenance, function and regeneration of peripheral nervous system. Schwann cell can be categorized into two major classes, myelinating and non-myelinating cells. Satellite cells. Satellite cells are precursor to skeletal muscle cells and are responsible for the ability of muscle tissue to regenerate. That is, these embryonic cells remain in the adult and can replace damaged muscle fiber to some degree. Satellite cells. It is precursor to skeletal muscle cells, and are responsible for the ability of muscle tissue to regenerate. These embryonic cells remain in the adult and can replace damaged muscle fiber to some degree. Concept of neural cells. Concept of neural stem cells. This is a new topic, concept of neural stem cells. Recently, scientific studies shows some nerve cells in the adult brain have the potential to divide and regenerate new neurons where the need arises. The cells may be termed as neural stem cells. These cells are capable of migrating to distant places where they may rapidly divide to give rise to new nerve cells. These new nerve cells send out long axonal and dendritic projection which integrate into pre-existing neuronal circuits and contribute to normal brain functions. This is concept of neural stem cells. And last we see summary. The nervous tissue is composed of neurons, nerve cell, process and neuroglia. These cells have a typical pale staining achromatic nucleus, dark nucleus and niche granules in the cytoplasm. Neurons usually have short, multiple, branching dendrites. The proteins produced in the cell are transported to the distal region of the axon and back. This is known as axonal transport. The classification of neurons is based on their structure, on their size, and on their function, like sensory neurons, motor neurons. Classification of neuron is based on their structures like multiple, bipolar, on their size Golgi type 1 and Golgi type 2, and on their function sensory neuron, motor neuron and interneurons. Neuroglial or glial cells are supporting cells of nervous tissue. They also produce insulation to neurons and their process. Ependymal cells help in the formation and circulation of the cerebrospinal fluid. The blood-brain barrier is formed by astrocytes while microglia are involved in phagocytic activity within the central nervous system. So this is about nervous tissue. If you have any doubt, you ask question and make a comment. Thank you for watching. Like, share and subscribe